Yeah, welcome to my presentation today. First of all, I would like to thank the organizers for giving me the opportunity to speak here today. This is a great honor. The title of my talk is Molecular Communication Fundamentals, Testbeds and Research Direction. This is the result of joint work with many colleagues, and some of them are listed here on this slide. So those are mostly my former or current PhD students that contributed to this talk. Then most of this work was conducted under the framework of a larger project, a so-called Emerging Fields Initiative of my university. And these projects are inherently interdisciplinary. And to give you an idea of the character of the project that we had, I have listed here the participating professors. And you may not know the names per se because they're from different areas, not from communications. But you can see the diversity of the team. So we had a colleague from the university hospital, colleague from chemistry and pharmacy, material sciences, biology, of course, also electrical engineering, mathematics, and it was a very, very broad team, and certainly our work has greatly benefited from this, especially the experimental part of the work. Now here's an outline of the talk. So this talk was designed to basically provide, on the one hand, a gentle introduction to the entire field of molecular communications to students or colleagues that may not have a prior background in this area. And on the other hand, I also tried to include some new work that maybe even those of you who are experts already have not seen before because it was just recently published or is still under consideration. So after a general introduction to the topic, then I will look at three different stages of molecular communication system design, namely at first the modeling part of a molecular communication system. And here we will focus on the channel particularly because of the time constraints. Then we will look at a concrete communication design problem. And this is in particular intersymbol interference mitigation. And we will see how nature basically deals with this issue and see how we can harness these natural ways of interference mitigation in synthetic molecular communication systems. Then we will talk a little bit about two test beds that were developed at my university. One based on a biological system and the other one based on a more synthetic approach with magnetic nanoparticles. Finally, we'll have the summary and the brief conclusions. Now, what is molecular communications? So molecular communication is supposed to enable the communication between so-called nanomachines. And the defining feature here is that the information is encoded in the concentration, type, or time of release of molecules. So in the properties of molecules, basically. When we say here nanomachines, we really mean machines whose components have sizes on the order of a few hundred nanometer. The entire machine, which could, for example, be a cell, is typically a bit larger than, is more in the micrometer scale. These nanomachines, they might be biological, right, as we said, a cell, for example, or they may be non-biological, such as nanosensors. And an example of such non-biological nanomachine you can see in the figure that is shown here. Right? So here essentially what we have here is a cut through a blood vessel. So we look inside a blood vessel. These round objects are depictions of red blood cells and these objects that look like spaceships a little bit. They're in fact tiny nanosensors that patrol this blood vessel. And inside this blood vessel, these nanosensors, they may, for example, look for biomarkers for certain diseases, such as cancer. And when they detect such a biomarker, then they may report this back to the outside world, basically. These nanomachines, they may get instructed to destroy diseased tissue, right? Like to attack cancer cells. For both approaches, so for both mechanisms, for the sensing part and the actuation part, of course, some form of communication with the nanosensors and maybe also between the nanosensors is helpful and necessary. And this is exactly a scenario where molecular communication could be helpful, right? So where the information might be embedded in the release of molecules, and this may help these nanosensors to carry out their tasks. Of course, this is a very futuristic concept that may take many, many years to realize, and there are many steps that we have to take in between, but this sort of gives you the long-term goal of part of this research. Now, molecular communication is prevalent in natural communication systems, so in biological communication systems. This is where the idea originally came from. And our goal is basically to harness this to build synthetic systems and also to better understand natural systems. So for the design of such molecular communication systems, at least 3 steps are needed, right? So are essential. One is the modeling part, right, that I mentioned in the beginning. So we need accurate models in order to be able to carry out a meaningful system design. Once we have these models, then we can go and design communication functionalities, synchronization, detection, and so on. And finally, we need a proof of concept, and to this end, we need to build test beds. And all 3 of these aspects will be covered in part in this talk. Now, before we have a closer look at molecular communication itself, let's first get a feeling of the order of magnitudes that we deal with here. So, what objects are really on the order of a nanometer in size. As a point of reference, something that you're familiar with is human hair, and human hair has a diameter of 40 to 120 micrometer, right? So it's much larger than a nanometer, but nevertheless, biological cells, surprisingly, are on the same order of magnitude, right? So biological cells have typically sizes between 1 micrometer and 100 micrometer, so they are really on the same scale as a human hair. Organelles, which are the organs or subunits of cells, they have sizes of 25 nanometer up to 1 micrometer. And on the right-hand side here, you see an artistic depiction of a cell, and the colorful objects inside, those are depictions of organelles. And these organelles have different tasks, and they also communicate with each other. Now, single atoms, they typically have sizes under 1 nanometer. And small molecules, such as oxygen, they have sizes of about 1 nanometer. Proteins, which are macromolecules that are prevalent in biological systems, they have sizes on the order of 5 to 10 nanometer. And viruses, which I'm sure you have heard about a lot over the last year, they have sizes on the order of 100 nanometer, right? So this just gives you a perspective of size in the context of biological systems as they are also relevant to molecular communications. Now, here in this picture, you see a scenario for a natural molecular communication system. In particular, you see here a colony of bacteria. So each of these rod-shaped objects is a bacterium. And these bacteria, they adjust their behavior according to their density or according to their number. So if there are only few bacteria in the environment, then the bacteria typically remain passive. and try to grow in number first. And then once they exceed a certain number, then they start to change their behavior and attack the host system. If we are the host system as humans, then this is the point in time when we fall sick, basically. Now the question is, how do these bacteria figure out how many other bacteria are in the environment, right, in order to be able to change the behavior? And the process by which they do this is called quorum sensing, and this is a molecular communication process. What happens there is that each individual bacterium emits signaling molecules, so-called autoinducers, and each bacterium also has receptors for the same type of molecule with which they can detect these signaling molecules. Now, if there are many autoinducers in the environment, then of course this means that there must be many bacteria that release them, and the individual bacterium will detect many autoinducers and then deduce from this, okay, there are many of us and it's time to change the behavior. On the other hand, if there are few bacteria, then there will be few autoinducers in the environment, and the individual bacterium will also detect only few autoinducers and not change the behavior. So this is one example of many examples for molecular communication in nature. Now what I just described is a form of diffusive molecular communication where the transmitter and the receiver is a bacterium. And this is one form of molecular communication in nature, and this is depicted here in the first figure, right? So the transmitter here might be one of these bacteria, and this here is another bacterium. These are the autoinducers, and the bacteria, they communicate with each other. This is not the only form of molecular communication in nature. Another form is shown in subfigure B here, and this is communication via so-called gap junctions. These gap junctions are small channels that connect the cytosol of two cells in tissue, right? So these rectangular objects that you see here is an abstraction of a cell, and what happens is that one cell produces signaling molecules. These signaling molecules can propagate to the neighboring cells via these gap junctions. In response, then these neighboring cells also produce the same type of signaling molecule, and then even more signaling molecules propagate to the next cell, and so on. So in this way, basically a wave of signaling molecules can go through the tissue, and this allows neighboring cells to coordinate their behavior. One example for this would be calcium signaling in tissue that uses this approach. Then in subfigure C, we see an example for molecular communication via molecular motors. This is, for example, used for the communication between two organelles inside a cell. And here, this is sort of a form of wired communication where the transmitter and the receiver are connected via a rail, a so-called microtubule. And along this rail walks the molecular motor, which has a vesicle attached to it, and this vesicle contains the signaling molecules, and the two legs that you see here are actually called heads and a head part and a tail part, and the heads, they're able to walk along this rail while consuming chemical energy, and in this way larger molecules and larger amounts of molecules can be transported from a transmitter to a receiver. Now the final form that I want to mention here is shown in subfigure D, and this is molecular communication via bacterial motors. So here a bacterium is used to transport signaling molecules from a transmitter to a receiver. With this approach you can achieve directed communication because bacteria can be attracted to the transmitter and the receiver via a food source and then pick up signaling molecules, for example a DNA molecule, at the transmitter and transport it to a receiver. Right? So this is another form. Now the one form that is prevalent both in the molecular communication literature and also in nature is actually the communication via diffusion, right? So the first kind of molecular communication that I mentioned, and therefore this is also the type that I will focus here in this talk. So now let's try to map this molecular communication to a conventional communication system sort of block diagram. Right, so from communications we know that we can actually depict any communication system or model any communication system in this form, where we have a transmitter and a receiver. In between is what we call the environment, or the communication environment, or the communication channel. And the transmitter has a message that it wants the receiver to know. Right, so this is the channel setup. And if we map this now to our molecular communication system, then the transmitter might be a cell, right? The environment would be the diffusive channel, and the receiver might be another cell. And the molecules, they move in this environment by, for example, free diffusion, or supported also by flow. We will discuss this a little bit more in the following. And in this way can convey information from the transmitting node to the receiving node. So in the simplest case, we may assume that the transmitter has a binary message to deliver, right? So of course in nature the messages are not always binary, right? Often they are, right? So it's just that either a signaling molecule is present or not, and sort of an on-off behavior. But there are also other examples where it cannot be easily mapped into a binary message. But let's assume that it can be mapped into a binary message, and in this case, for example, the transmitter may emit a certain number of molecules whenever it wants to send a binary 1, and not emit any molecules when it wants to send a binary 0. Now if it wants to send a binary 1, right, then these signaling molecules are released at the transmitter, they propagate in the channel, and some of them will arrive at the receiver. Receiver has receptors on its surface, and these receptors can bind the signaling molecules, and if enough signaling molecules are bound, then the receiver will sort of, or the receiving cell, for example, will change its behavior, as in the example of the bacteria, or some internal processes in the cell will be set in motion, and this can be interpreted as a threshold, as a detection process, right? And since we have to observe a certain number of molecules in order to get this started, this might be seen as a threshold detection. So in many cases we basically model this just as a threshold detection at the receiver. Now let me say a few words about applications. So the type of application that was originally envisioned for molecular communications were nano applications, so where we use molecular communication in environments that are very small, where the conventional electromagnetic based communication cannot succeed. And examples for this are of course medical applications like targeted drug delivery, where drugs are released collaboratively, for example in the cardiovascular system, or for health monitoring, where biomarkers and toxins are sensed also inside the body, right? So this is very much in line with the picture that I showed you in the beginning of these small nanorobots that patrol a blood vessel. Now, the general concept of this was also coined the Internet of Bio-Nano-Things, and essentially this is sort of the networked human being, where we have a sensor network, an actuator network inside the body that is based on molecular communication, and that is connected to the outside world via gateway nodes to a conventional communication system, and then allows us to basically do these personalized type of healthcare applications. The same concept, of course, can be transferred to other areas, for example, to the environment, right? So where also we may want to detect and degrade pollutants at a much smaller level than what is possible today, right? So we can detect the pollutants much earlier when the concentration is still low, before they actually become a problem, and then degrade them. And similarly, for quality control, we might be able to identify product defects at a much smaller scale before they actually become visible and problematic, right? So this is very much in this nano domain of applications. This was the original idea, but then of course over time people realized that it will take a long time to realize these nano applications, right? So there are many obstacles in between that have to be overcome. And therefore they also started to look at goals that are maybe easier and more in the short term to realize. And for this, of course, it would be convenient if we could do away with the nano aspect of it. And this led to macroscopic applications, or the search for macroscopic applications. And here several proposals were made. Mostly they tried to benefit from the sort of different properties of molecular communication systems, and that allow these systems to operate in environments where electromagnetic systems cannot operate, such as in liquids or in explosive environments. So an example here would be monitoring of oil and gas pipelines, right, where you could do the monitoring inside the pipeline by adding some signaling molecules to the oil or to the gas that is traversing these pipelines anyways. Then in industrial plants where explosive gases are used, this might be useful, right? Because there you may not be able, or you may not be allowed to use electromagnetic waves for communications because of the explosiveness. Or in aircraft turbines where the rotating metal parts cause a problem for electromagnetic communication, but are actually beneficial. The flow that is generated might be beneficial for molecular communications. And finally, it may allow you to control the chemical process more effectively, because you can actually control the process inside the medium where the process happens, and you may not need an extra sensors that convert the signal into an electrical signal first. So those are the macro applications. Of course, they are a little bit hampered by the fact that usually molecular communication is slow at large scales, right? So it's not as fast as other forms of communication. And therefore, in the long term, the more promising ones are certainly the nano applications. Good. So then, if these applications have such great potential, the question is, of course, why do we not already have such molecular communication systems? And the answer is, of course, that this is still in its infancy, right? So the research into this, and several things are required which we do not fully have, right? So on the one hand, what we need is, of course, accurate but still simple communication theoretical models for the transmitter, the receiver, and the channel. We have such models for conventional wireless communications, right? So we characterize the channel as Rayleigh fading, Ricean fading, right? We know for a cable how the channel is modeled, but we do not know it really for many of the practically relevant molecular communication channels. So this is one aspect that we need to solve. Then once we have this, we need to develop the functionalities of such communication systems. And these include, of course, things like modulation, detection, channel estimation, and so on. But the way this might have to be done at the nanoscale may be very different from what we are used to at the macro scale, right? And I will show you one example for this today, actually. And then finally, we have to verify our concepts with experiments, right? And this is maybe the most difficult step, because as electrical engineers, we may not have the full background to do this, right? The competence to do this, and therefore we need to collaborate with colleagues from other fields. As I said, in this talk, we will mostly focus on channel modeling, then mitigation of intersymbol interference, and then some experimental verification. Before we go into more details, let's have a quick look at how this research area has evolved over the last few years, right? Because those of you who are new to this may not be aware of this. You may say it can be traced back to 1959, where Richard Feynman gave his famous lecture, "There is plenty of room at the bottom." And this is often seen as the spark that started basically nanotechnology. Of course, he did not talk about molecular communications, but since molecular communications is sort of inspired by nanosystems, this played certainly a certain role. Then there are many examples in the literature in recent and more distant years where actually communication theoretical and information theoretical concepts were used to explain biological effects, for example, in kidneys, in a paper by Johnson and Knudsen in 1965. But there are many other examples for other systems, right? But this is not really molecular communication, because in molecular communication we want to do the opposite, right? So we actually want to build synthetic molecular communication systems with the help of biological concepts, right? So it's sort of the other way around compared to what traditionally has been done. Then the term molecular communication has been mentioned by Toby Berger in his 2002 Shannon lecture. But the term molecular communication in the context as we use it here, so basically as a synthetic communication system built based on concepts that can be found in nature, this was probably first mentioned in a conference article by colleagues from Japan in 2005. Then in 2008, ComSoc started an emerging technical initiative on nanoscale molecular and quantum networking. Such initiatives are sort of working groups inside the IEEE, and they are started whenever a new topic sort of emerges on the horizon where communication may play a role, right? So this was the point at 2008. Then in 2010, Elsevier started the first journal on nanocommunication networks, and this journal is still in operation today and largely dedicated to molecular communication. In the subsequent years, most of the communication conferences that we know, right, Infocom, ICC, Globecom, they started workshops or symposia on this topic. And in 2012, ComSoc also started a series in JSEC, so in the Journal of Selected Areas in Communication, on this topic. And this series was later converted to a separate journal in 2015. This became the journal on the IEEE transactions on molecular, biological, and multiscale communications. In 2013, already the first standardization group was founded on this topic, right? So, and this is still in operation. Also, in the same year, a textbook appeared on molecular communication, the first textbook by Eckford and Nakano, and I think now they're already at the second edition of this book. Then in 2014, the ACM, so this is sort of the counterpart of IEEE for computer scientists, they started the ACM Nanocom conference. So this is a separate conference that mainly deals with molecular communication, at least with nanoscale communication, and is an annual conference that also happens every year. In 2019, the proceedings of the IEEE had a special issue on molecular communications. Of course, the proceedings is a very prestigious journal that cuts across all areas of IEEE. And when they do a special issue, this means usually that the topic that is dealt with has already reached a certain maturity, right? So at least has been around for a few years and also shows great promise, right? So this is sort of the requirement. And they had such a special issue on molecular communications. And finally, in 2019, the ETI, so this Emerging Technical Initiative that I mentioned before, actually graduated to a technical committee. Technical committee is a more permanent structure within the IEEE, and this is a working group basically now on molecular, biological, and multiscale communications. So from this you can see that it already has a history of I guess now 15 years or so, this area. Still many new aspects and new journals and new conference and so on get added basically almost on a yearly basis, and there's a lot of excitement around this topic, but it's still new and still many new aspects. Okay, so then let us delve a little bit deeper into the whole area of the modeling of molecular communications, since it's quite different from the modeling that we're used for electromagnetic communication systems. And the basic mechanism that happens there at this small scale is diffusion. And at the level of an individual molecule, this means that the individual molecule carries out a so-called random walk. So this is a random a random movement that is caused by thermal vibrations and collisions with other molecules, right? So you see this here for an example, right? So the straight line is the movement, but then the molecule bumps into another molecule, and as a result, it changes the direction, and this gives the appearance of this random movement. Now, this random walk is fast over short distances, but it's quite slow over longer distances. And while the movement of the individual molecule is random, its statistical behavior can be described by mathematical equations, basically. So this can be predicted, and at the same time, this also would predict deterministically the behavior of many molecules, right? So for this, there's a very rich literature on diffusive processes, because this diffusion plays a role in other areas as well, such as heat transfer or fluid dynamics, that are much more established than molecular communications. This literature can be exploited, of course, and should be used for modeling such systems. Now, the basic equation there is this Fick's diffusion equation, this Fick's second law, and this is shown here. Here, this Fick's diffusion equation basically describes the evolution of the concentration of the molecules, and C here is the concentration, R is the space variable, and T is the time. So this CRT tells us what is the concentration of the molecules that we are interested in at point R at time t. And how does it change? Well, on the right-hand side, you have the change with respect to time, right? So the first derivative with respect to time, and on the left-hand side, and on the right-hand side, you have the second derivative with respect to space, right? So this is the Laplace operator here applied to this concentration, and d is the proportionality constant. This is called the diffusion coefficient. The larger d is, the faster the diffusion happens, right? So the faster the movement. Now, this is a partial differential equation, and it looks very simple, of course, but if you want to solve it actually for a given environment, it turns out that this can be quite challenging. And this is because the solution of this partial differential equation depends on the boundary conditions and the initial conditions of your environment. So the equation that you will get for the concentration of the molecules at a certain point in time and space will depend on the shape of the environment, right? So whether you basically are inside a blood vessel, so you have sort of a tube or rod-shaped environment, or inside a cell, right? So can be maybe modeled like with a sphere, or you have an unbounded environment. The same, of course, it depends on how the molecules are released, right? So is it a point source, or are they released from a sphere? And also, are they released instantaneously, or are they released slowly over time, right? All of this will affect how how this concentration changes over time and space, right? And it turns out that for many, or actually for most environments, even the practical ones that we deal with in biological systems, there's no closed form solution, right? So there might be an analytical expression for this concentration, but often it is quite involved and involves infinite sums and so on. So not that insightful, but yet useful to derive, because it may give you at least allow you numerically to evaluate the concentration relatively quickly, and we'll see examples for this a little bit later. Now, there's one exception where actually the expression that you get for the concentration of the molecules is quite neat, and this is the special case where we release the molecules instantaneously, right? So we release n molecules instantaneously from a point source. And there are no boundaries in the environment, right? So this would correspond to the case where you have a drop of ink. You put this drop of ink in the middle of the ocean, right, at a certain depth, and then see how the ink molecules develop over time and space, right? So how they move over time and space. This would be basically an example for this scenario. And in this case, you get a very simple expression for the concentration of the molecules, which is given here. Right, so where n is the number of molecules, d is the diffusion coefficient, t is the time, and r is the distance from your point source. Right? And what do we see? Well, you see it's a simple equation, but with respect to time, we will see that it first increases and then it decreases the concentration. And with respect to space, right, with respect to the distance from your source, you have here the r square in the exponent. Right? So this means that you have a decrease over distance. So the further you're away from the source, as we would probably expect, the fewer molecules you will observe because the concentration of the molecules is lower. Now, this simple expression is used in many publications on molecular communications, right? So actually most papers, I think, still use this as the basic model because it's easy to understand and it's simple. But, you know, we have to keep in mind that it represents an environment that typically does not happen in a practical system, right? Because there are no boundaries and we have an instantaneous release. If you make this model only slightly more challenging, for example, we have now a two-dimensional model with just two boundaries, right? This is the only change, so we introduce two boundaries. Then the corresponding concentration of the molecules is given by this equation here. And the details are not so important, but you can see that here we already have an infinite sum, right? So if you compare these two, then you see, you know, this here is a lot less insightful compared to this one. And this is, you know, not a very complicated environment. It's just the addition of boundaries, right? This will do this to our concentration. And I guess this gives us an idea of the challenges that we face here, right? So it's difficult to get insightful analytical models, basically. But let's go back to the first equation that we had, right? So to the simple one and illustrate this a little bit. So we plot here this concentration as a function of time, right? So basically we observe the number of molecules at a certain distance from our point source after the point source has released the molecules. And we observe it at different distances. So the different curves are for different distances between transmitter and observation point. And what we see is that, of course, first we do not observe any molecules because, you know, it takes for the first molecules a little bit to arrive at the observation point. But then the concentration quickly increases at our observation point, and after that it decreases again because the molecules, they will diffuse away into infinity because there are no boundaries, right? We also see that the larger the distance, you know, the fewer molecules we observe. Now this characteristic that we have there, so the concentration at a certain point in time, the concentration at a certain point in space as a function of time after impulsive release, this is also called the impulse response of the channel, right? So this would be the impulse response of this molecular communication channel for a point receiver and you know, the instantaneous release. Right. So this is the, you know, this gives you an idea of how the concentration develops over time. To make this even clearer and also to add a little bit more reality to it, I actually want to show you a video for such a diffusive channel. We keep the transmitter as it was, so we have a point source transmitter and an instantaneous release, but we make the receiver slightly more realistic. So instead of having just a point receiver that observes the concentration, we actually try to model a cell. And our cell is a sphere which has receptors on its surface. We model these receptors for simplicity as triangles. And if a signaling molecule hits one of these triangles, or one of these receptors, then it gets bound to the receptor with a certain probability, which is specified by a so-called forward reaction rate constant. And once it is bound, it may get unbound again, so the binding is reversible with a certain probability, which is characterized by the backward reaction rate, right? And maybe we just have a look at this video. Right, so here you see the sphere, you see blue triangles, and you see gray triangles. The blue triangles represent that area of the cell that is covered by receptors. Of course, not the entire surface can be covered by receptors, so the gray triangles, they represent the part that is not covered with a receptor. So if a signaling molecule hits the gray part, then no reaction, so the molecule cannot get bound, right? And therefore it will just get reflected from the surface. If it hits a blue triangle, then it may get bound with a certain probability, and then may get released again after a while because of the backward reaction rate. Now, of course, in reality, these receptors are not triangle, right? So they may have more of a round shape, right? More of a circle type shape. However, for the modeling, it's much easier to deal with triangles, right? For our simulation. And also, what really counts is more the area, right? For the statistical behavior, and not so much the exact shape. Okay, so the transmitter is hidden actually behind this button, and now I click this button and release the signaling molecules, right? So now you see our signaling molecules, these red dots, they move randomly by diffusion. And here one has activated a receptor, and when the receptor is activated, then the counting may start, right? So then a signal might be generated inside the cell that basically gives rise to some process, and this is what we interpret as detection. You see here we release many signaling molecules, but only few of them arrive at the receiver because there's no direction to this, propagation, right? So they move in all directions. But you also see that we do not need a line of sight, right? So here actually we activated a receptor on the back side that has no line of sight connection to the transmitter, and this is again due to this random movement, right? Now, this would not be a very effective system because the distance between transmitter and receiver is relatively large, and the unbounded environment sort of make the diffusion inefficient, right? But I think just to illustrate how this works, for this it's a good, it gives you some intuition behind, first of all, the propagation of the molecules, but then also what a practical, what a realistic receiver may look like and what it may do. Okay, so then let's get back to this. Right, so this was diffusion. Of course, diffusion is not the only mode of transportation in molecular communication environments. In fact, there are some environments where diffusion does not play a huge role, right? Where actually another phenomenon is more important, and that is flow, right? For example, in the cardiovascular system, right, there's the blood flow due to the pumping of the heart. And in this case, for most processes, actually the flow is the aspect that transports the particle and not so much the diffusion. Now, for the flow, there are different types of flow that might be distinguished, namely uniform flow, laminar flow, and turbulent flow. Uniform flow is the one that has been mostly studied in the molecular communication literature because it's the easiest one to deal with analytically. And it assumes that the flow velocity is the same across the entire space, right? So it can be described by a three-dimensional vector with fixed components that are not dependent on space and time. Now, if this is true, right? So if you have this uniform flow, then you can still use the same expressions for the concentration as before. All you have to do is you have to replace the distance r with an effective distance, which is given by this equation here, which now not just depends on the point in space x, y, and z, but also on the flow velocity vx, vy, and vz. Okay, so this is the good news. The bad news is that there is no really, usually this type of flow does not occur. And it would only occur if, again, you would have an unbounded environment. But usually you have boundaries, and if you have boundaries, then the flow can be characterized either as laminar or turbulent. The exact flow profile that you will get there, so the exact velocity profile, you can obtain from the so-called Navier-Stokes equation. So this is another partial differential equation now for the velocity field. In general, also very difficult, and usually you can only solve it numerically, except for a few simple geometries. For example, for a tube-type channel, right? So those are actually geometries that we often encounter in practice, right? So a tube is, you know, a model for a blood vessel, so this is realistic. And for this case, you can calculate the flow profile, and there the flow profile is parabolic, right? So the velocity is has a parabolic distribution across the cross-section of such a tube, meaning that in the middle of the tube you have the highest velocity, and at the boundary of the tube you have zero velocity. Now if you just have this laminar flow, then, you know, things are actually fairly simple, and you can model the channel quite effectively, analytically. However, usually you have an interplay between this laminar flow and diffusion. So usually you have both of them at work. And if you have both of them at work, then things become complicated again. And I've illustrated this a little bit here in these three figures. So we assume here that our particles are initially uniformly distributed here across the cross-section. So this is actually a three-dimensional tube, right? So with a circular cross-section. And we cut through this tube, and then what we see here is the cut, right? So we see just what happens at the cut that we make. So this is the initial distribution of the particles, and then the flow comes and, you know, moves them along. And then after a while, the distribution, if you just had flow, would look like this, right? So there you see this parabolic shape of the velocity profile. So the particles that are closer to the boundary are further behind because they move with a slower velocity. And those that are at the center, they move faster, so they are ahead, right? So this is exactly this model. Now this is when the flow dominates, right? So the diffusion can be neglected. So either the flow is very large, is very fast, or the diffusion, coefficient is very small. For the case where both play a role, diffusion and flow, then of course what happens is that this here is superimposed with diffusion, and therefore this is what you see in the next figure, right? So here you can see now these particles, they start to move up and down and forward and back, right, due to diffusion. So it's not clearly such a parabolic shape anymore. And in the final figure, the diffusion becomes even more important, and now you can see that we cannot even see that there was a parabolic shape anymore. And you have to take into account both. Now for the general case of this interplay, there is no neat analytical model, right? Does not exist. Only for the flow-dominated regime there is a model, right, which then just uses this parabolic shape. And in the asymptotic case, so if the system can develop for a long time, then sort of an equilibrium between flow and diffusion happens. And this can be described by the so-called Aris-Taylor regime. And it turns out that in this Aris-Taylor regime, so if you wait long enough basically for the particles, for the diffusion to fully kick in and affect the particles that are moved due to flow, then you can model this equivalently with a uniform flow, right? So with a uniform flow profile and an effective diffusion coefficient, right? And I will show you actually a video for this where you can sort of see this in the next slide. Okay, so the videos that I will show you now are for the same scenario that we just saw in these figures. So we have a straight tube with laminar flow and diffusion, and we release the particles either from a point source or uniformly, right, in different videos. And what I would like you to look for is, on the one hand, the comparison between a flow-dominant regime and a diffusion-dominant regime, right? So how different the particle movement is there. And also the contrast between a uniform release and a point release, right? So how this affects actually the particles that we can observe at a receiver at a certain distance. Okay, so... Okay, so let's start with the uniform initial distribution, right? So again, this is the cut through our tube, right? So this is the set. This set direction is the direction in which the flow goes, and the yellow line that you see here, this is the initial distribution of our particles. So they are uniformly distributed across the cross-section, and because of the cut we just see a line there. And now we start the flow process, right? And you can see, due to the flow, how the particle concentration changes over time, right? And you can clearly see that the particles at the center, they move faster than those at the boundaries, right? Those directly at the boundaries, they do not move at all, actually, right? Or hardly, because there's a small amount of diffusion still present. And those in the middle, they are far ahead, right? You can also see that this actually causes dispersion, right? Because now, if you would observe these particles at a certain point in space, you would not just observe them at one instant in time, but you would observe particles over a period of time, right? Because the particles get spread out in space due to this laminar flow. Okay, so this was the first. The second one is the same scenario, right? But now we consider a case where diffusion is more pronounced, right? So either, the flow is the same. Here in our case, actually, the flow velocity is the same, but the particles are smaller such that the diffusion coefficient is larger, so they diffuse more. And here you can see what happens. So in this case, you can hardly see the parabolic shape, right? What you can see is basically that the particles collectively move from left to right. Right, so the yellow area really moves from left to right still, because there's still flow present, but at the same time, this looks like a uniform flow really, right? So the flow velocity seems to be the same across the entire cross section, and the broadening of the concentration, right? This is due to the diffusion, right? Because here diffusion is important, and therefore our flow profile broadens. And therefore, this looks really, this is really this Aris-Taylor regime where your combination of laminar flow and diffusion looks like the combination of a uniform flow with the same velocity, flow velocity everywhere across the tube, and diffusion. So those were for uniform releases. Now let's contrast this to a point release, right? So here the particles at the beginning are concentrated in one point. Not exactly a point of 0 dimension, right? So it's like distributed in a small sphere, but nevertheless in a very small space. And then we release them. And clearly in the flow-dominated regime, what you can see is that the initial distribution of the particles is sort of preserved. It just gets a little bit spread out due to the flow profile that we have. And you can imagine that this moves sort of along this parabolic flow profile, right? So the particles get spread out a little bit in space, but they remain at the same y coordinate roughly, right? At the same height here, and diffusion is not important, right? So basically, when you observe these particles at a certain distance, you would still know that they were initially released from a point source, right? Or you would suspect this. This is very different if you have a point source, but you're more in the diffusive regime, right? So now same initial conditions, but now we are more in the diffusive regime. And in this case, you can see that quickly the initial particle cloud broadens, and after a while, what you see there, right, looks very much the same as for the uniform release in the diffusive regime. Right? So at this point here, it's not obvious anymore that we release the particles from a point source rather than with a uniform release. Right? So we have completely forgotten the initial conditions, which means that essentially, again, we are in this Aris-Taylor regime, and the particle distribution looks the same regardless of how you release them in the beginning. Okay? So what I want to illustrate with this is that the interplay between flow and diffusion leads to an overall transport mechanism that is not really obvious, right, and has to be studied really case by case, right? So this is the key observation here. Okay, so now we covered uniform flow and laminar flow. Of course, there's also turbulent flow. Turbulent flow becomes important if the liquid moves faster, and also if there's some turbulences introduced from the outside, right? So it's more likely to happen in an air-based environment, right, rather than in a liquid, and it's more likely if the flow velocity is faster or if there's some obstacles in the environment. Now, I do not cover it here because there's much less literature on this, has been covered much less in the molecular communication literature compared to the other types of flow. So this would be a good topic actually for future work, the turbulent flow. The third phenomenon that I would like to consider here is the degradation of signaling molecules in the channel. So not all signaling molecules that are released remain in the channel. Some of them, they are degraded. Either they degrade by themselves, right? And with degradation we mean that they are converted to a different type of molecule that cannot be recognized at the receiver anymore, right? One mechanism that helps this along is actually the degradation via enzymes, right? And this is found, for example, in nature in the neuromuscular junction. So this is the gap between a neuron and a muscle cell. There the neuron stimulates the muscle cell with neurotransmitters, so with signaling molecules via molecular communication. And it does so by first releasing these neurotransmitters, so the muscle gets stimulated and contracts. But then, of course, you also would like to release the muscle again, right? And this is done by subsequently releasing enzymes that degrade these neurotransmitters into a different type of molecule that will not stimulate the muscle cell, right? Modeling-wise, this is illustrated here, right? So A here would be our signaling molecule. E is the enzyme. If an enzyme and such a signaling molecule, which is also called a substrate in this context, if they come close to each other, then with a certain probability they will bind, right? And this is characterized by this forward reaction rate K1. Once they're bound, they form this, the compound EA. And then two things may happen. Either they separate again without effect on the signaling molecule, and this happens with a certain probability that is characterized by this backward reaction rate. And with another probability, the enzyme will actually convert the signaling molecule to a new type of molecule, to an AP molecule that cannot be recognized at the receiver anymore. The enzyme goes out unharmed from this process, right? So then the enzyme in the next cycle can basically convert another signaling molecule. Now, this chemical reaction here, this can be modeled by the so-called Michaelis-Menten equations. And this then overall leads to a reaction diffusion equation, right? Actually to a set of such reaction diffusion equations. So basically to coupled partial differential equations, which are also nonlinear. So therefore, we cannot solve them in closed form, right? These reaction diffusion equations in general. But often what is possible is to find an accurate approximation. And we did this actually in our prior work. So we found such an approximation for a scenario that is sort of typical for molecular communication environments. And what we found was that now the concentration of the signaling molecules can be described in this manner here. So this is the same equation that we had before for the unbounded environment, right? So the blue parts that you see in this equation are exactly the same. The only thing that is now new is the red part, and this red part is due to the degradation with the enzymes, right? And you can see what this does to the equation. So without this red part, for large T, this concentration basically as a function of time behaves like a polynomial, right? So you have a polynomial decrease of the concentration over time for large enough times, because this term here becomes 0, and then this term here dominates. Now with this red part, this behavior changes completely, because now this part here actually increases with time, and therefore you get an exponential decrease of this impulse response, or of the concentration for large times. This is also visible here in this plot, right? So where the dashed line is for the case without enzyme, and the black curve is for the case with enzyme, and you can see with enzyme we have a much faster decrease, right? So we also have a smaller peak, right, which might be undesirable because some of the signaling molecules get degraded before they arrive at the receiver, but nevertheless we also have a much faster decay. Okay, so this was the third phenomenon that I wanted to mention here in the context of channel modeling. Now, what you've seen from my presentation is that there are many effects that in the channel that we cannot model accurately, right? So where we need approximations, right? So this was already apparent for the diffusion alone, right? The equation became very involved quickly, and we needed probably approximations. Then for the flow, right? For the laminar flow and the interaction with the diffusion, we probably need approximations, and then finally for this degradation also we needed approximations. And therefore, of course, it's important that we can verify that these approximations are accurate. And ideally we would do this with an experiment, but experiments are expensive and we cannot always carry them out. Therefore the next best thing is a simulation, right? So therefore we need to verify this with simulations. And we identify here on the slide 4 different ways of doing simulations, and they roughly go from a very coarse approximation to a very fine approximation, or a very fine simulation of reality. So the first example is a continuum simulation, and there what you basically do is you solve in one way or another these partial differential equations numerically. And an example for this would be the second set of videos that I showed you, right? So where we saw how the concentration of the particles moves through the channel, right? So this would be an example for such a continuum simulation. And this does not give you statistical information about the individual particle. This just gives you sort of the average behavior of the signaling molecules. The next best option is mesoscopic simulations. And there you divide your entire space into smaller subspaces, right? Like small cubes, for example. And you basically, describe what happens inside such a cube, again, now on a probabilistic level, right? So with what probability do the 5 particles that are in cube 1 move to cube, how many of them statistically would move to cube 2, or how likely it is that a chemical reaction happens in a particular cube. This is called a mesoscopic simulation. Then the third one would be a microscopic simulation. Those are typically particle-based simulations where you track individual particles. So you track their random walk movement, and you track how they move due to the flow, and you also track individual particles and see if they're close enough together for them to basically engage in a chemical reaction. An example for this was the first video that I showed you, right? So where we had this sphere with the receptors on the surface, and we saw how the particles moved with random walk, right? So this would be an example for a microscopic or particle-based simulation. The last one is the molecular dynamics simulation. And this molecular dynamics simulation is a very accurate simulation that actually also simulates the forces between particles, right? So this is not like we just simulate them as a point, but we basically simulate or model every element of the particle. So if it's a protein, then every atom on the protein, how they are connected, what forces are active between them, and then what forces as a result are active between different molecules. So this is very fine, has a very fine granularity, has a very high complexity, of course, right? So roughly you can say the complexity increases from A to D, right? So the models become more accurate and more complete, but also more complex. These molecular dynamics simulations, they're mostly used to by biologists to model the interaction between, for example, a receptor and a signaling molecule, right? But just one interaction, right? So, for example, for our enzyme and signaling molecules, maybe with this we could just model the interaction of one enzyme and one signaling molecule, but the complexity is too high to model an entire communication system, right? Okay, so now these simulations are important, and actually in the literature and also online you can find a number of simulators already. One very convenient simulator is this Accord simulator that was developed by Adam Noel, my former PhD student who is now a faculty at Warwick University. And you can download the simulator from this GitHub page and also play a little bit around with it. So with this you can get the type of simulation that you saw in my first video, right? I mean, how the individual particles move. This is what you can base in different environments. This is what you can do there, and you can build your own system, basically your own communication system. Okay, so with this, I think I complete this modeling part, and now we have a quick look at a design problem. And this design problem here is intersymbol interference, and I picked this because all of you are probably familiar with intersymbol interference from your wireless communication courses. What is it? Well, typically we saw before that if we send just one pulse of, if the transmitter releases molecules one time, then we get this type of pulse, right, this dashed line. But often in communication systems, we want not just to send one symbol, not just one bit of information, but another bit a little bit later. And therefore, we have to release a second pulse of molecules, which arrives at the receiver in this form. And now you can see that there's overlap between these two pulses. And this overlap is what we call intersymbol interference. And you know from maybe from conventional wireless communications that this will lead to less reliable communication and more decision errors. So what can we do to overcome this? Of course, the simplest option is to increase the spacing between the pulses, because if we increase the spacing, there's less overlap, so there's less interference, right? However, this will decrease the data rate, of course, right? So this is the downside of this. Then we have very effective mechanisms to deal with this in conventional communication systems, and there what we do is we have at the receiver a device that can undo this intersymbol interference or exploit it, right? So either by maximum likelihood sequence estimation or by some sort of filtering. And this is of course a good option if you have a macroscopic molecular communication system where you can afford the complexity. But at microscale, this might be difficult to do, right? So you may not be able to teach a cell how to do this Viterbi algorithm. And therefore, it might be too complex for nano receivers, okay? So it pays to basically see how nature deals with this, right? So can we get some inspiration from nature? Because there also, this phenomenon should happen, right? So intersymbol interference might be also a problem there. And indeed it is, right? And we already had a glimpse of this. Namely, one mechanism that is used there is these enzymes that reduce the excess signaling molecules, right? I already mentioned before this neuromuscular junction, and here you have a depiction of this, right? So this here would be the end of such a neuron, and this here is the muscle cell, and the space in between is this neuromuscular junction. And there we said before that for the muscle cell to relax again, what the neuron will do, it will also release enzymes that basically degrade the neurotransmitters that are still in the environment. This allows the muscle cell to relax and then to send another pulse at a later point in time, right? So this would be one way of dealing with this intersymbol interference in a molecular communication context. Another option is actually to remove the signaling molecules by reuptake, right? And this is what happens in so-called tripartite synapses. There the molecules are uptaken either by the presynaptic neuron or by surrounding cells, so-called glia cells. So here you have a sort of a model for this. So this here is the presynaptic neuron. This here is the postsynaptic neuron. And when an action potential arrives at the presynaptic neuron, right, so electrical signal, then this information is basically transferred to this postsynaptic neuron by release of neurotransmitters, so signaling molecules. And the signaling molecules are here these green dots that you see. So they're released by the presynaptic neuron, and the postsynaptic neuron has receptors. These are these black circles here, so illustrated by these black circles, that can bind these neurotransmitters, very much like what you saw in my video, where the cell basically could, the receptors on the cell surface could bind these signaling molecules. The same happens here at the postsynaptic neuron. They are bound, and then after a while they are released again. And the surrounding area, so this here is the blue surface. This illustrates the membrane of so-called glia cells, right? So those are cells that surround this synaptic cleft in these tripartite synapses, and both the glia cells as well as the presynaptic neuron, they have proteins embedded in the cell membrane that can uptake, so that can remove these neurotransmitters from the synaptic cleft. So if a signaling molecule hits such a protein, then it will get absorbed either into the presynaptic neuron, and there it might get recycled and used again later for transmission, or it gets absorbed by the glia cells, right? And this absorption of the signaling molecules, of course, this also decreases intersymbol interference, as we will see a little bit later, because the molecules are removed from the environment, right? Simply. Okay, but let's first have a look at the enzymes, right? So the last two approaches I would like to discuss here a little bit more. Let's start with the enzyme, right? So we saw before we have a transmitter, we have a receiver, and now we have the enzyme in the environment. The transmitter releases the signaling molecules, and some of these signaling molecules, they get degraded by the enzyme, right, as we saw before. And as a result, the impulse response gets shortened, right, between transmitter and receiver. And this allows us to avoid intersymbol interference, right, because now the shorter impulse responses, if we transmit at the same rate as before, they do not overlap, right? Or if we can live with some overlap, then we can actually increase the data rate, right? So we can space these pulses closer together, right? So this would be one way of dealing with intersymbol interference. The second one is, as we said, this uptake, right, at this tripartite synapse. So here the presynaptic neuron releases the neurotransmitters, right? They propagate here via diffusion to the postsynaptic neuron. They get bound, right? And they may get released, so this binding is reversible. At the receptors, they make it released again, and then if they hit either the presynaptic neuron or the glia cells, then they can get uptaken and removed from the environment, right? Now, to model this mathematically, basically we model this synaptic cleft here as a three-dimensional cuboid. This is an approximation, of course, right? So the real shape is different and basically difficult to model, but this gives us a first-order insight into what happens. And we have to model the receptors here. You know, we cannot model them individually. Instead, what we have to do is a boundary homogenization and model this as a reversible adsorption at a homogeneous surface. But then we can, in our simulation, of course, we can model the actual receptors, and we compare the simulation with the analytical result, and you know, we will see that this is actually very accurate. And then basically the presynaptic neuron and the glia cells, they are modeled as partially absorbing or reflective boundaries, right? And then with these ingredients, you can formulate a fixed diffusion equation and try to solve it, which we did. The actual equation that you get for the impulse response is a little bit involved, right? So not surprisingly, there's an infinite sum. But for large enough t, the impulse response becomes quite simple. So you can approximate it with this equation here, right? So where beta and alpha are simply constants. and d is the diffusion coefficient. So in other words, for large enough times, we can see that we have an exponential decrease of the impulse response between the presynaptic and the postsynaptic neuron. And alpha and beta are constants that depend simply on the on the parameters of the system. And you have this illustrated here. So here we investigate the impact of two parameters, namely the desorption coefficient at the postsynaptic neuron. So this basically is a measure for how fast the signaling particles are released again by the receptors after adsorption, right? So after binding, how fast can they unbind again? The larger this coefficient, the faster the unbinding happens. And the reuptake rate, here Kr is the kappa r is the other parameter that we use. The larger this reuptake rate is, the more likely it is that a particle that hits the presynaptic neuron, that it's actually uptaken again. So for the figures I show you, we assume that the glial cell boundaries are reflective. So let's first look at the family of these three curves, which have one desorption coefficient, which share the same desorption coefficient, for different reuptake coefficients. And what you can here clearly see is that the larger this reuptake rate, right? So this green curve has the largest reuptake rate. This has the smallest reuptake rate. The largest the reuptake rate, the more particles are removed, right? Or the faster they are removed from the environment, and the faster our impulse response decays here, right? So you can clearly see this fast decay. I should say that the dashed lines that you may see here, this is this asymptotic formula, and you can see this is very accurate for the decay, right? Not accurate for the increase here of the impulse response, but very accurate for the decay. The solid lines are the exact solution, right, that I do not show, and the markers that you see are the simulation results. So you can see that the approximations that we make here with this boundary homogenization is actually very, very accurate. For the second set of curves here, you see a similar behavior. Here, Kd, the desorption rate, is larger, which means that particles that are bound to a receptor at the postsynaptic neuron, they are released faster again, right? And since they are released faster, therefore at any time, any point in time, on average, fewer particles are bound to the postsynaptic neuron, and therefore, in general, here the impulse response is smaller. Right? So this is what we can observe from there. In general, what we found analytically was that this alpha, right? So this tells us how fast the impulse response decreases. This alpha increases with the uptake rate, kappa r, of the presynaptic neuron and the unbinding rate of the postsynaptic neuron, right? And of course, a larger alpha means a faster decrease, right, of the impulse response. And alpha decreases with the binding rate at the postsynaptic neuron, right? So a binding rate means that how easily it is for them to bind to a receptor at the postsynaptic neuron. And the distance between presynaptic and postsynaptic neuron, right? So this distance actually plays a large role in how fast the impulse response decays, because the distance basically determines how likely it is for a signaling particle to hit the presynaptic neuron or also to hit the glial cells. Okay, so this was this part on intersymbol interference mitigation. My main point here was really that the problems that we encounter in molecular communication systems are very much the same as those in macroscopic systems, but the way to overcome them might be very different from what we are used to, right? So in conventional communication systems, we try to overcome intersymbol interference at the receiver, whereas in these molecular communication systems, actually they are usually mitigated by the channel itself, right? So either by adding enzyme or by having a reuptake mechanism at the boundaries. Okay. Now, the next point here is the experimental verification, and here I have actually a quote by Richard Feynman, and he said, "It doesn't matter how beautiful your theory is, it doesn't matter how smart you are, if it doesn't agree with experiment, it's wrong," right? And the point that I try to make here is that a large part of the work that we've done so far in molecular communications, right, globally, I mean not just my group, but most groups, was very much focused on developing theory and concepts. And of course, this is also important, right? So it's important, it's an important step. But at some point, these theories and concepts, they have to be actually verified by experiments. And this is really where the largest challenge is in this area, and where the most promise probably also is. So to give you an idea of what such experiments could look like, I want to report a little bit about the two test beds that we have here at FIU. The first test bed is a biological test bed, and is basically optical to chemical signal converter. It exploits so-called bacteriorhodopsin proteins, and those proteins, they occur in nature in the so-called halobacterium. And the property that they have, this bacteriorhodopsin, is that in response to light stimulus, they will emit protons, right? So therefore, in this natural environment where they occur, they will emit protons into the environment, and therefore there's there are more protons at the outside compared to the inside of this bacterium. And the concentration gradient that happens, of course, nature tries to equalize this. Therefore, these protons, they try to move back into the cell. And they can move back into the cell via other proteins. And these other proteins, however, they are sort of molecular machines that use the work that is conducted by them allowing to move back to generate or to convert this work into chemical energy. And they use this to generate ATP, right? So basically, this is used by these bacteria to convert optical energy into chemical energy, right? That's the original purpose. Now we, of course, modified this. So what we did is, or I should say my colleagues from biology, from Professor Burkowski's group, what they did, they genetically modified E. coli bacteria such that they also express these bacteriorhodopsin proteins on their cell surface. And with this, of course, now you have a modulator, right? Because you can shine light on these genetically modified bacteria, and they will emit the protons. The protons are then our signaling molecules, right? And they can be detected at a receiver, and with this, as we will see, you are able to send, as a proof of concept, simple 0-1 messages, right? So basically, this is sort of an interface between a macro and a micro molecular communication, a macro scale and a micro scale system, right? So really an optical to chemical modulator. Here you see the setup in our laboratory, right, or in the laboratory of Professor Burkowski. This is a schematic, right? So our transmitter basically is this light source, right? So with this we can basically modulate the light source. The light source then modulates here the bacteria in the suspension. Our receiver is an electrical pH sensor, and then the pH value, right, is connected to the concentration of the protons, and therefore we can measure the concentration of the protons, and this is our received signal here, right? So this is the setup. Now, of course, we have to try to model this, right? To understand it, we have to model it and use the things that we've seen before for modeling and maybe also for communication system design. And the model that we came up with for the concentration of the protons at the receiver is given here. It has 4 different components. So the first one is just the reference concentration at time 0. So, for example, at the beginning of a simple interval. X of t is a deterministic signal due to the illumination, right? So this is our useful signal basically that we send. dt is a drift term, and E is a noise term. So the last 2 terms sort of are due to the properties of the bacteria, so they are impairments. Now this x of t, this deterministic signal, for this we could show that it has sort of an exponential relaxation behavior. Right, so maybe we look at this example here, right? So this is the optical signal, right? So we switch it on, we leave it on for a while, then we switch it off, right? So this is we use on-off keying basically. And the deterministic part of our signal, this x of t, that we get in response and that we determine analytically, would be this signal here, right? So you have first the increase, right? Then this is the pH value. Sorry, this is the concentration of the protons, right? So it goes up, it stays up, and then it goes down after we switch the light off again, right? So this would be x of t here. However, you can see that the blue curve here, this is the measurement result, the fast moving blue curve, this does not really fully agree with the black curve. So there are some additional effects that we could not take into account in our model. In particular, one term was this drift term, right? So we saw that in the long term, this proton concentration changes over time no matter what we do, right? And this is due to effects that, as I said, is outside our model, and this may include, for example, that these bacteria that we use, they age over time, and then they change their behavior, and they become less effective, or some of them die, so the number decreases, or also they may proliferate, right, and basically create more bacteria, and this may also influence the signal. So this, basically, we saw that we can model by a linear drift term, right, this dt. And then, of course, there's always noise, right? So the quick movement is due to noise, and this could be either diffusion noise or measurement noise or just the noise from our pH sensor. If we take all of this into account, then we can see that the red curve here, which is our theoretical model, agrees actually very well with the measurement result, which is the blue curve, right? Then we extended this, right? So we wanted a proof of concept. So we sent basically a sequence of zeros and ones, right? So here this is the optical signal, right? So where the optical signal is switched on and off then for a while. So this is the sequence that we want to send. And this is the corresponding chemical signal, right? So now in this case it's directly the pH value. And the blue curve that moves more quickly that you see, this is the measurement data, right, that we determined directly measured with a pH sensor. And clearly you can see that on the one hand, well, there's a drift, right? On the other hand, you can also see that whenever we send a 1, clearly the pH decreases. Then when there's a 0, it increases, then it decreases, and so on. So this seems to be really effective, right? So the information that we have embedded in the optical signal seems to be reflected also in the chemical signal. And you see there's a good fit between the theoretical model, which is the red curve, and the analytical result, right? So it's a very good agreement. Now, as a final proof of concept, we actually want to extract the information that was embedded in the optical signal from the chemical signal again, right? So we want to build a detector. And this experiment is shown here, right? So here, this is what happens whenever the where you have a yellow line here. This means that we switch the light on, so we send a binary 1. Where it's white, we send a binary 0. And this is the corresponding the corresponding chemical signal that we observed, right? The corresponding pH value. And then in order to build a detector, of course, we could go and build a maximum likelihood detector for this, and we did, right? However, there's also a very simple way of dealing with this, and this is to just try to combat the impairments that we have, right? And we saw that there are two types of impairments. One is the drift and one is the noise. Now the drift changes at a much lower scale than our desired signal, right? So there's a very long-term effect. So you can get rid of this, you can eliminate it by applying a high-pass filter, right? So you apply a high-pass filter that filters out the drift term, and here we used in particular a differentiator, right? So we differentiated the received signal. On the other hand, the noise changes at a much faster scale than the desired signal, so therefore you can filter it out with a low-pass filter. Now this combination of high-pass and low-pass filter gives the signal that we observe here that moves somehow fast, right? So the black signal that you see here. And then after this differentiation, you can just use a simple threshold detector to distinguish between zeros and ones. And the red line is the threshold that we used. And at least in this case, we could really detect the ones and the zeros from this received signal again, right? So this basically was a proof of concept that yes, this molecular communication based on a biological system is possible. even though, of course, it required a lot of work, right? Much more than what I could present here. So, these bacteria you have to cultivate, right? You have to genetically modify them, you have to cultivate them, you have to keep them alive, right? You need the right temperature, you need, everything has to be right. They age over time, so, there are many things that we learned from this. That also one of the takeaway messages was that we had for us was that, such biological systems are very difficult to deal with. And, if there are alternative ways to build a test bed that you can use for experiments, then, this might be also worth to explore, right? One that is not so dependent on biological components. And this motivated us to think of a second test bed that is actually based on magnetic nanoparticles. These magnetic nanoparticles are so-called iron oxide particles. And what made them attractive to us are two things. So first of all, their size is variable and typically in the nanometer range. So they're in the same range as molecules, size-wise, and therefore they behave similar as molecules as far as transport is concerned, right? So that you can study diffusion, flow, and all of this also with these nanoparticles. And then second, they are iron oxide particles, so therefore they're paramagnetic, so you can actually, on the one hand you can attract them with a magnet, but on the other hand you can also easily detect them because of this, because they're iron oxide particles. And this was a second attractive feature. Now, one nanoparticle is not exactly one iron oxide molecule or compound, but a collection of them, right, that are, they typically agglomerate together in this core, in this iron oxide core. And then in order to prevent more of these compounds to stick together, usually they are coated, right, with some polymer material. And this then altogether, this is the what we call then the nanoparticle, right, the magnetic nanoparticle. And this coating is important to prevent more particles from sticking together and from agglomerating, right? So to really have a similar behavior as for a molecule, you need this coating. Now these particles, they're already used in nature, right? There's a little bit, in the medical field, as illustrated a little bit here. So they are used for drug targeting, for imaging, and for tissue engineering, right? So for one usage cases, for example, they are functionalized with cancer drugs. So this means that cancer drugs are attached to the surface here. And then they are injected close to a tumor, and then a huge magnet is used to hold them close to this tumor, right, by exploiting their magnetic properties. And this has been shown by our colleagues here from Professor Alexios' group at FIU to be much more effective for cancer treatment compared to the conventional treatment where you just inject the cancer drug in the bloodstream and hope that it will get to the right point in your body. With this, you can do it in a much more targeted way, of course. Now, for us, the main benefits are that they are readily available, they are stable, right? So you can buy them. For us, Professor Alexio, of course, fabricated them, and they're easy to handle. The test bed that we built, you can see here. So the purpose here was to study the interaction of flow and diffusion, mostly flow, as it turned out. So this here is the channel that we had. So you can look at this, and this is sort of like a part of a blood vessel, if you want, right? We generated a background flow here with one pump, right? And then we had another pump that injected here the particles. So this here would be the transmitter part where we inject the magnetic nanoparticles. And this here is the receiver. This receiver is a so-called susceptometer, which generates an electrical signal in response to the presence of these nanoparticles in the core of this coil, basically. And with this, you can send, again, simple binary messages as a proof of concept, right? So here you can modulate this pump, right, how it pumps in the particles, and then here you can basically record the signal and you can send a simple message. This here is the setup in the laboratory, right? So this is an older setup. By now we have a more sophisticated setup. This setup here uses, custom off-the-shelf components, right? So this here would be the susceptometer. This here is the pump for the background flow, right? I think this is the pump that injects the nanoparticles. Those components you all can basically buy, and maybe if you have colleagues that work in this area, they may already have them, right? So that work on fluid dynamics or on modeling blood vessels, they may already have such equipment that you may be able to use for your work. Now, of course, our main interest was to use this to verify models for molecular communication. And there we saw that one important component is the flow, right? So we have this background flow. So for the flow, we saw before there are different regimes, right? There's on the one hand, there's the flow and there's the diffusion, right? So first of all, we try to figure out how important diffusion is compared to flow. This can be done by the so-called Peclet number, right? And we found that this Peclet number is quite large, which suggests that this is a flow-dominated regime. So therefore, in our model, we neglected diffusion, and we only assumed that there's laminar flow there's flow in the channel. Then, of course, you also have to see whether the flow is actually laminar or it might be actually turbulent. And there's another number in physics, the so-called Reynolds number, that helps you decide this. And based on this, and the elements that go in there are like the flow velocity and the distance and the width of your tube and so on. And based on this, we found that the flow really should be laminar and not turbulent. In other words, the channel itself can be modeled as a tube with a laminar flow, right? So therefore, this is illustrated here. This would be the flow profile that we should have in the channel. Here you see a picture of where we release these nanoparticles, right? So this here is the main channel. This is where the particles are injected, and this is again the main channel, right, that provides the background flow. And you can see here that this has sort of this laminar flow profile, right? You can see that here some particles are very fast, right? This is in the center of the tube, and here they are slower, and this is more at the boundaries of the tube. So you can really see a little bit this laminar profile. Now the receiver here is easy to model because the susceptometer basically counts the number of particles that are inside the tube in a certain area, right? So this is very easy to model. It's like what we call a transparent receiver in molecular communications. What turns out to be challenging is the transmitter, right? I mean the particle injection here, because here we have this pump that injects the particles, but while they're injected, they already moved through the tube, right? So this is difficult to handle analytically. So therefore, we came up sort of with an approximation for this. And in our approximation, we model the transmitter as if the particles, after injection, are only distributed across the cross-section here, right? So they're all located in one point in Z direction in this cross-section. And across this cross-section, we came up with a parametric model for the initial distribution of the particles, right? And this is given here, this expression. The details are not so important, but this has two parameters, this distribution, alpha and beta. If you choose alpha equal to beta equal to 1, then this simplifies to the case where the particles are all uniformly distributed across the cross-section, right? So basically, at the circle, the two-dimensional cross-section, right? So the circle, they are uniformly distributed inside the circle. This is what happens if you choose alpha and beta equal to 1, right, for the beginning. And if you change the parameters, you can generate different initial distributions that basically reflect how fast you pump and how many particles you pump into your main tube, right? Now, if you use this model and combine it with the laminar flow and the receiver model for the susceptometer, you get a simple equation for the channel impulse response, which is given here, which depends, of course, on these parameters alpha and beta, then on the distance between transmitter and receiver, and this u is the flow velocity, right? So this is the peak velocity at the center of the tube. This is valid for times larger than d over u0. So, and this initial delay that is introduced here, this is, for the time that basically the particles at the center of the tube need to reach the receiver, right? That's why you have here first U of t is 0, but then after this time the first particles will arrive and will be counted. For large t, the decay that you get for your impulse response is 1 over t over beta, right? So you have a polynomial decay. And of course, such a parametric model is useful, right? Because it helps you for channel estimation. Because here you only have to adjust a limited number of parameters and fit them to your measurement data. And this is then a channel estimate that you can use, for example, for detection. Now, these two figures here illustrate a little bit the impact of the initial distribution on the impulse response. So here we have two different cases for the initial distribution on the right-hand side. And on the left-hand side, we have the corresponding impulse response. And what you see is basically if you have a number of particles close to the center of the tube, right, as is true for this orange and blue curve, then you have a peak for the impulse response right after the delay, right? So here, once the first particles arrive, you get a peak. This is due to the particles that are here at the center of the tube, right? So this here is the distance of the particles from the center of the tube, right? This is this rho. This is the distribution of the particles, right? So this is one thing. For the green curve, we do not have particles close to the center of the tube, right? Therefore, here you have a much gentler increase of the particles that you observe at the receiver because there are no particles at the center at the beginning. And then we also see that the more particles we have here at the boundary of the tube, right? So one here corresponds to the boundary of the tube. The slower your impulse response will decay, right? So here for the blue curve, this is the extreme case where we have actually quite a few particles close to the boundary initially. And these particles, they move very slowly, and therefore here you have a very slow decay of the impulse response, right? The blue curve decays much slower than the other two curves. So therefore what we see is that this initial distribution, right, so how you inject the particles, this has a huge impact on how your particles propagate subsequently. And this is also something that you saw in the video, right? We saw in the video in this flow-dominated regime, there was a big difference between how the particles moved through the channel between the uniform distribution at the beginning and the point source at the beginning. Okay, but now how does this compare to our measurements, right? So this is shown here. So here, the gray curves that you see, those are sets of measurement results for 4 different distances, right? So for a distance of 5, 10, 20, and 40 centimeter between transmitter and receiver. And the colorful curves, right? So the red, green, and so on curves, those are the curves that the analytical curves that we fit to these measurement results based on adjusting the 2 parameters that we have, right, alpha and beta, and the measurement, right? So we did a curve fitting basically. And you can see that actually the fit is very well, right? Of course, the individual impulse responses, they will deviate a little bit from the curve fitted version, but still they're close enough, right? So we actually get a good fit. And we also see that the corresponding initial distribution, so if we use the parameters that we got here from the curve fitting and plug them into our initial distribution, then this is the initial distribution that we get. And therefore all distances close together, right? So they're almost identical. And this is, this is intuitively pleasing because, of course, we would expect that the initial distribution of the particles does not depend on when you observe them finally, right? So these curves should really all be identical, and they're close together, I mean, for experimental result, basically, right? What you also see is that for the way we inject the particles here, we have few particles at the center, right? And most particles are here around, are not so far from the boundary, actually, right? So therefore, a higher injection speed that brings them closer to the center, or a slower one, right, if we already passed the center, right, because this just tells us the distance. This does not tell us where exactly we are with the particles, right, whether we already passed the center or we actually have not reached the center yet, might be advisable, right? I mean, it would be desirable to have a faster decay of these impulse responses, and therefore it might be good to look for injection mechanisms that have more particles at the center as opposed to the boundary. Nevertheless, is a good fit with the measurement results. Now here I show you some more measurement results for the received signal. Maybe we concentrate on the blue curves here in these 4 sub-figures. The blue curves are measurement results for transmission of a sequence of symbols. So we transmit here ones and zeros. In each case the same sequence is transmitted, but we measure the received signal at different distances. So again, from 5 to 40 centimeter. And what you see is basically that the larger the distance between transmitter and receiver, the more intersymbol interference we will have. This means that the signal that you observe will be smoother and smoother, right? So here for 5 centimeter, you clearly see still when a 1 and a 0 was transmitted. Here you see this less, right? Because of this effect of intersymbol interference that is caused by the longer impulse response, right? You also see this here, that for 40 centimeters the impulse response is longer compared to here the 5 centimeter length, right? Then we did an experiment where we actually counted errors for detection schemes, right? So we sent 300 data symbols. We sent some initial symbols potentially for channel estimation, and then we sent 300 data symbols, and we detected them after 5, 10, 20, and 40 centimeter. We used on-off keying and counted the errors. Of course, this is not, not statistically significant because we have only 300 data symbols. Nevertheless, it gives us an idea, right? I mean, of where this is going. And we compared 3 different detection schemes. The first one was a simple increased detection scheme. For this detection scheme, you just pick 2 points in your symbol interval, and you look whether the signal within the symbol interval has decreased or increased, right? And based on this, you decide whether a 0 or a 1 was transmitted, in the simplest case. And we see from the results here that this is actually quite effective, right? So this does not need any channel estimation really. And, we have some errors for 5 centimeters, but 0 errors for 10 centimeters and few errors then for 20 and 40 centimeters, right? So it's a suboptimum scheme, therefore the number of errors you make does not necessarily linearly monotonically increase with the distance, but in general it works reasonably well, I would say. And then we compared this with 2 sequence estimation schemes. So for sequence estimation, you need to know the channel impulse response, so you need to estimate it, and we estimated it either based on our model, right, that we presented before. So we just had to estimate 2 or 3 parameters, right? So we only needed very few training samples. And we also estimated the channel impulse response with a sample-based scheme, right? So this is the regular estimation scheme that is also used for wireless communication, so sort of a least square error scheme. And in this case, you need at least L-1 training symbols, where L is the length of the impulse response, right? So there we needed much more training symbols compared to the model-based scheme. And then in both cases, we used these impulse responses and put it in a Viterbi decoder to basically get the error rate. And what do we see here? Well, we see that the model-based scheme actually works fairly well, right? However, the sample-based scheme works better for larger distances, and this is probably due to the fact that this model-based scheme has probably some mismatch, right? There's some model mismatch, whereas the sample-based scheme does not have such a model mismatch, even for larger distances. But of course, the downside is that you need much more training symbols. Also surprisingly is that this increased detection actually works as the best scheme for 40 centimeter, right? Where for these large distances I would have assumed that the sequence estimation schemes are far superior. And they probably are, but if everything is ideal, so if you know the impulse response perfectly, but due to the estimation error that we have invariably, right, especially for these larger distances, this destroys sort of the optimality of the sequence estimation again. Okay, so this sort of was supposed to illustrate that actually these systems can be realized, even though the realizations that we have today are maybe still a little bit away from the actual application that we envision for such molecular communication systems. But nevertheless, this is a first step into the right direction, I think. And in recent years, or say in the last year, really we could see several groups in the world that started to do experiments and that publish their results. So I think this is still a very promising area. Now this brings me to the end of my talk. So I would like to give you a brief summary and an outlook, really quite short here. So I hope I could convey the message that molecular communication is really a new and quickly evolving area. You could see a little bit that it's inherently interdisciplinary. This makes it challenging, of course, to enter, right? But this also makes it very interesting, at least to me, in the long term, and holds great promise. Within IEEE ComSoc, we have a very active and rich community, actually, with a technical committee that deals with this, and we have meetings at every IEEE and GlobeCom conference, and everybody is welcome to attend these meetings. Also, we have symposia at all IEEE major ComSoc conferences, right? So at ICC and GlobeCom, there's always a special track for molecular communications, and finally we have now a dedicated journal for this, the IEEE Transactions on Molecular, Biological and Multiscale Communication. So ComSoc invested a lot in this area, and it starts to bear some fruits. Then the basic message that I also have is that to really get a little bit familiar with the processes in nature, right, and try to harness some of these processes and exploit them for the design of these molecular communication systems. I find this very, very rewarding. Now, as the outlook, well, it's a young research area, right? So there are many open communication and information theoretical problems, right? So this is what, as I said, makes it interesting to me, right? It's, in many aspects are quite different from regular communication systems, and usually we like to think in Gs, right? So in generations of communication systems in wireless communications. So we say, 4G, 5G, now we are doing research for 6G. Well, this research is maybe not for 6G, but for 10G or for 20G, right? So this is very forward looking. But the differences are huge, right? So I mean, if you compare the differences between 4G and 5G, right, and you have to explain it to a layman, you can say, okay, in 4G our base stations had 8 antennas, and now in 5G they have 100 antennas, and therefore the system works better, right? So I mean, this is a little bit a simplified view, of course, but it's not that far from the truth, right? So if you tell this somebody outside our area, they probably will laugh at you, right? Now here, this is completely different, right? So here, this is really groundbreaking, and has no, it's not an incremental change. It's a paradigm shift, basically. Well, more work is needed, right, to advance the field, of course, and my advice here would be to try to get together with your colleagues from biology, fluid mechanics, medicine, and neighboring areas, right? So I do this too, and I always found this very rewarding, and with this you can learn a lot in a short time, basically. And in the end, of course, we need applications, right? So when you saw my summary at the beginning of the history of the field, and now also what I explained about the ComSoc community, this is very much still an academic issue, right? So it's still very much an academic community. In the end, we need industry support, right? So we need applications that are attractive to industry. And in order to develop such applications, we need a close interaction with them, right? So we need to try to draw more people from industry in this field, and then, every one of us in our own research try to find areas that may have, a more direct impact on products. Yeah, I think with this I come to the end, and I'm very much looking forward to your questions afterwards. Maybe before I show you a few related publications that we have on which this talk is based. I don't want to go really one by one through them, just show them briefly, and of course also we can make this the PDF file for this talk available And so then you can check this out for yourself. But until then, thank you very much for your attention. And as I said, I'm looking forward to your questions and to answering them. Thank you very much.